All right, so let's take a look at Chapter 28. Chapter 28 is a survey of protists and all the different groups and types of protists that there are. Remember that there's controversy about how to break this group up. Originally, there was, the whole group was just a place where if an organism didn't fall into plant, animal, or fungus, then they would just throw it in here. And what scientists have been starting to realize is that actually all of these different organisms that are in this group called protists are very dissimilar from one another and share different or do not share a common evolutionary history. And so, because of this, they are looking at breaking this up into multiple kingdoms, but nobody's really quite so sure on how to do it because sometimes the DNA evidence can contradict, lifestyle evidence or the organelle structure evidence. And so, there is a lot of controversy about how to break this group up. Um, and you'll see that as we're going through this survey. Now, keep in mind that many of the different groups that we're going to talk about here, there's a lot that you could potentially know about each of these groups, but I want you to focus on what sort of makes each of these groups different from each other. Kind of the two major points that you would say this is what differentiates this type of protist from this type of protist. Trying to remember everything is just going to take you forever and cause you a lot of heartache. So there are three basic overall or sort of general classifications that we give for protists. There are the ingestive protists. These are the ones that eat things, and they are more similar to animals. We've got the photosynthetic protists. They are more like plants, and a good classic example of them is algae. And then you've got the absorptive protists, which are similar to funguses and display some of the characteristics that would make you think, well, this is more similar to a fungus than an animal or a plant. So these are the three overall groupings that we have, and you'll see each of these types of lifestyles in all of the groups that we see coming up. So those are the three groups of protists that exist. Protists are generally speaking unicellular organisms, although they sometimes do come together to form multicellular life. A good example of that could be the brown algae, which form kelp forests. So that's a good example of multicellular life that is a protist. But most of them are unicellular, and a lot of them have sort of interesting lifestyles that we call them mixotrophs, meaning that they mix characteristics like photosynthesis and heterotrophic nutrition. So we talked about that in the last PowerPoint, that being heterotrophic means that you get your carbon from another organic source, but at the same time these protists do photosynthesis. So we call these interesting protists mixotrophs. They're mixing different ways of making a living, and they're really, really important in the ecosystem. Examples are plankton and phytoplankton in our oceans, which are the foundation of all marine life. So without protists, you couldn't have large whales, you couldn't have, the complex ecosystems that currently exist there. So they're important to us in one way as scientists because it helps us understand, how do you get to animals? How do you get to plants? How do you get to funguses? But it's also important on a practical level that really, all the marine life is supported by protists and their activities. So the endosymbiotic theory, which we covered in the last section, helps explain the diversity of protists. Remember that at some point in eukaryotic life's history, mitochondria and chloroplasts found their way into the eukaryotic cell. And mitochondria came first, chloroplasts came afterwards. You're going to see that this is important for different groups of protists because some protists have different types of mitochondria or different types of chloroplasts. and that helps us delineate one group from another. So remember that all protists are eukaryotes. Some of them are single cellular, some of them are multicellular. One of the things that differentiates a lot of the different protists is the types of plastids that they have. A plastid is just a pigmented organelle, and a chloroplast is an example of a plastid. So one of the things that's created these unique organelles inside of protists is something that we call secondary endosymbiosis. So in primary endosymbiosis, we think, as you just saw on the last slide, that some photosynthetic bacteria entered a eukaryotic cell and it would eventually become a chloroplast. And once you have the formation of these organelles, then we can have the formation of things like red and green algae. So red and green algae are both protists. They're both very closely related to plants, but they themselves were engulfed by other eukaryotes or were parasites of other eukaryotes and eventually led to the formation of a number of different groups of protists. So up at the top you can see how when red algae became engulfed by some of these other protist life forms, they formed a different type of plastid organelle, and that caused the differentiation that we see up here in the top right hand corner. So each of these different groups of protists has a different type of plastid, and all the plastids originate from red algae. So how this sort of red algae came to be inside of these cells tells us that the differences there are what separates these protists. You've got green algae also coming into some types of eukaryotic cells, and there's a symbiotic process that goes on here. What is the algae getting out of this? Well, it's getting protection from potential predators. And what is the host getting out of this? Well, it's getting the energy from the photosynthetic activity of this plastid, or what used to be a green algae. And so it's really a mutualistic relationship, and over time, based on how these, basically when and how the evolution of this symbiotic relationship occurred, we have the differentiation between the group euglenids and this other one down here. I want to say. So basically, as just like sort of final summary here, what you've got is not just one symbiotic event, but two symbiotic events, and this is what has led to different groups of protists being found. So there are a couple groups of protists that have modified mitochondria. And by modified, we typically mean smaller. Some of them lack DNA, some of them lack electron transport chains, some of them lack enzymes needed to carry out the specific functions of cell respiration. And these organisms also don't have plastids, so they cannot go through photosynthesis. So a couple of these groups that have both modified mitochondria and no plastids are the diplomonads. And where they get their name from is the fact that they have two nucleuses and multiple flagella. So up in the top right hand corner you can see this. They have multiple flagella coming out of them. You can't see the double nucleus, but that's where they get their name from. How would you potentially know this? You may have heard of Giardia. I think I know some of you guys as students in the past. I've known a few of you have come down with this. And if you were wondering where you get that from, well, you eat or you take in drinking water that's been contaminated with feces that has the cyst or part of the reproductive stage of this organism inside of those feces. And so that's where you would get that from. and you would get that from going to, let's say, towns outside of Guadalajara where you might have unsafe drinking water. All right, there's also a group called the Parabacillids, and these are known basically through having vaginal infections. And the reason why these infections flare up is because you take antibiotics, it kills the bacteria that are naturally living in that environment, and without the competition, this type of protist explodes, and that's what causes the infection. So that's another group that has modified mitochondria neoplastids. All right, so there's a group of protists called the euglenozoans, and what's special about these guys is they have crystalline rods inside of their flagella. Nobody's quite certain why they exist, but they're there, and that's what makes them different from the other group of protists. These are further broken up into the euglenids, and what's special about euglenids is that they have sort of like a little pocket from which their flagella emerge. What's interesting about them is they can switch between autotrophic and heterotrophic activity, and they also have the ability to phagocytize. So going back to cells, remember, that's when you take in something large. Another group of euglenozoans are the kinetoplastids, and these are interesting because they have a large mitochondria with a central DNA mass called a kinetoplast. So, if we're remembering back to the endosymbiotic theory, when mitochondria come into the cells, maybe this is a mitochondria that retain more of its DNA in this group of organisms compared to other ones. And so that's sort of what makes it interesting. You may have heard of this one because of sleeping sickness. You've heard probably of before in various literature, or Chagas's disease. And the way that these work, the way that they're able to cause the symptoms involved with both of these diseases, is that they have proteins on their surface which are constantly changing to avoid detection from the immune system. Remember that the way that the immune system works is by trying to detect what's part of you and what's not you, what's foreign. And they can never really develop a proper defense against this type of protist because of that. All right. Some of you may have been asking the question, sort of what is sleeping sickness? And it's just a disease that's caused by the tsetse fly in Africa. And, the reason why it's called sleeping sickness is eventually you go into a coma and die if it goes untreated. And then you've also got Chagas's disease, and this is transmitted by blood-sucking insects, and it's common here in Latin America, and it can lead to heart failure. So that's what those diseases are. And at this point, when I close out the PowerPoint, I'm going to zoom in on the image of the top right of the euglenid so you can see its pocket. There you go. So you can see this pocket where the flagella is coming out of.